Great. Thanks for the introduction. Yeah, I'm going to be talking about something a little bit different. We're basically trying to bring the same concepts that work for the mycoplasma cell into a eukaryotic organism. So um, I'm Zach. I work in the Aaron Reich lab at USC here in LA. And we're basically trying to produce minimal chromosomes, eukaryotic chromosomes in this organism. Um, and this will be, of course, a refresher for everybody here, but minimization, basically, we're asking the question, what are the minimal genetic components that can sustain cellular life? And if we just think about a 6-gene organism that we know everything about, really we just want to get down to a point where we can't reduce the organism any further. That's the sort of basic definition of what a minimal genome might look like. When we think about eukaryotes, things get a bit more complicated. In Saccharomyces cerevisiae, the budding yeast, we have 6,000-ish protein coding genes. And we are fortunate enough to know that about 70% of those genes are essential. But it's relatively intuitive to understand that removing all of the non-essential genes isn't the way to actually produce a minimal organism. It's not likely that we'll be able to produce a yeast that actually survives just on that 17% of the essential genes. And to kind of illustrate this point a little, I'll go back to that toy model genome. If we know everything about this genome and all the interactions, we can start to take genes out of this organism. We can remove a single gene. And as a result, we end up with 2 of the non-essential genes being converted to essential because of the way that they interact with the removed gene. This is fine. We can continue minimizing this organism, but we have choices. We can end up deleting this gene here or the gene on the right over here. And these produce 2 distinct genotypes, one of which is actually minimal. As a result, we can't remove any further genes, but we still have some options on this lower one here. And when we remove genes there, we end up with a distinctly different minimal set of genes. And really, this is just to illustrate that when we expand this 3 orders of magnitude to 6,000 genes, our predictability of what a minimal genome or even a chromosome actually constitutes is very limited. And if we can't predict very reliably from existing data what that genome might look like, how do we actually produce and test this in the organism? So in our lab, our question kind of became, can we use some sort of natural biological process to do this sort of synthetic genomics design, build, test cycle to reduce chromosomes in vivo? Because it's not practical to either do these knockouts stepwise, because we're not really sure where we'll end up, or if we'll be limited very quickly. The number of options is so huge that producing all of them is not technically feasible. And then even among the options, very few of them are probably actually viable and minimal themselves. So we've been thinking about this question for a long time while we were working on a publication that came out in 2023 called Building Synthetic Chromosomes from Natural DNA, which I got to work on. In this paper, effectively, we clone natural regions from the yeast genome to build synthetic chromosomes from them using in vivo processes like homologous recombination. And as a part of that work, we tried to delete 10 non-adjacent genomic elements from the first chromosome in Saccharomyces cerevisiae. Effectively, what we do is we clone these regions in orange in vivo, take them out, and pop overlaps onto them, which we can use in vivo to then reconstruct the chromosome without the non-cloned regions inside of yeast. When we did this, we actually observed that all of the viable versions of this designed chromosome had retained this region containing a gene called SYN8. You don't need to know anything about SYN8. What's important here is that the retention is happening, and it appears that it's required for viability of the organism. And really, the mechanism at which this is happening is something that we wanted to leverage. We hypothesized that inside of cells, what's happening is that the synthetic clone segments can recombine with their native counterparts to retain the natural region of the genome into the final chromosome. And as a synthetic geneticist, this is actually a problem. You're not getting out what you designed for. But we've been thinking about minimization, so it started to actually feel like it might be a solution to the minimization problem, this sort of concept of allowing the cell to recover from designed deletions effectively. So the bulk of my dissertation I'm a fifth year now, for the past like 3 years has been developing this method that we're calling mosaic. Effectively, mosaic is just a method for doing exactly what we wanted to do, to experimentally reduce these chromosomes in vivo. And it leverages this sort of observation we made on that previous work. So the way mosaic works is we have, and I'll be talking about chromosome 1 in this case. We take candidate genes from that chromosome, which include all of the known essential genes on that chromosome, as well as a couple of other candidates, which I don't have time to get too deep into right now. But effectively, we synthesize all of these genes in red, and we order them as overlapping fragments so they can recombine with one another to construct a very small version of that chromosome, the putatively minimal chromosome. But when we do this inside of the yeast, what we saw before can occur. These fragments can recombine with the natural segments, sort of in the way that meiosis might work between a reduced copy of the chromosome and the native chromosome. And as a result, we get out these sort of intermediately minimized chromosomes and their native chromosome counterpart. So this is sort of the designing and building part of this across a transformation plate where we get many distinct clones out of this transformation. We expect that we get many different genotypes of this minimized chromosome. And effectively what we're trying to do is account for regions of the chromosome that are annotated as non-essential, but might become essential and be required in the context of these reductions. Now, to test them, we can eliminate the native chromosome from this population using its conditional centromere. And what we get out are cells that have reduced genotypes that are actually viable genotypes. And just as an example, if we look at these guys, we can go ahead and sequence all of our individuals with short read Illumina sequencing in something that might look like this. Sorry, this graphic is not loading. Something that might look like this, where we can get an idea of the genotype of each chromosome by looking at the coverage of each region within that organism. And you can see differing numbers of deletions across these two individuals. And we can display this in sort of a matrix format that might look something like this. When we do this experiment on chromosome 1, we did it about a year ago. We end up with a handful of different genotypes, which I'm showing here in this matrix. We've got all of the individuals here as the columns, and then the rows denote each possible deletion, which is just a window between those two gene fragments. And we get up to about 63% of the chromosome removed, both by base pairs and the ORFs that are present on the chromosome. And as we reduce the individuals, we can see this, the doubling time of these organisms tends to decrease with increasing reductions, which is kind of a strange way to put that, but we also increase the variation. We were pretty excited to see this result initially. I'll just talk about this genotype, which is the smallest of the chromosomes that we've produced so far in an individual called BA6. About 63% of the chromosome is removed, as I said before, and it has a pretty long doubling time, which makes it a little bit difficult to work with. I'm kind of pampered. Yeast is incredibly easy to work with, and so any reduction in it kind of can be frustrating. But we wanted to do a lot of downstream analysis with BA6 that we ended up doing with a different strain. BA6 qualitatively is just really difficult to work with. I don't have quantitative evidence for any of this, but it doesn't transform very well. It's not resistant to a lot of the antibiotics that we normally use, even when those genes are installed into the chromosome. And there are a couple issues with the actual ploidy of the chromosome 1 itself. So we moved on, kind of in what might be thought of as like an add-back experiment that was just produced naturally from this to this genotype, AC5, that has a few more genes. It has about 6 more protein coding genes, but it's much easier to work with. It's very similar to wild-type yeast as far as the workability goes, even though it grows slower. So for the rest of the talk, I'll be everything that's done is done in AC5. So we know now that mosaic can generate these reduced chromosomes, but the question becomes, how do we know that the chromosome is actually minimal? Like, what does that mean? Do we know that content on AC5 still can't be deleted? And then kind of as an aside, but I won't have time to talk about it, how does the minimization of this individual chromosome affect the content of the genome as a whole? And to kind of get at these questions, we used in vivo saturating transposon mutagenesis using this transposon called Hermes. It's an inducible plasmid-based system, which we can transform into AC5 and its parental cell, and then induce transposition and take a peek at what survives that process, which is very similar to what was done in the original minimal cell paper. So the best way to display this data is a little bit lost on me. Oh, and I should mention, we're now getting into pretty preliminary data. I actually got this back on Monday, so a lot of the interpretations that I'm making are kind of live right now. We got a lot of insertions. We got 200,000 unique inserts, which is outlined in this black, kind of in this circle plot of the genome. And then in AC5, we got 30K unique insertions. We had the same depth of sequencing in both strains, but AC5 seems to have, or appears to tolerate insertions much less than its parental strain, which is kind of what we might expect, but needs to be followed up to make sure that's not a technical artifact. So if we kind of dive into the chromosome 1, which is what we're interested in here, what I'm showing here is all of the genes on the chromosome. In blue, we have the known non-essential genes, and in red, we have the essential genes on that chromosome. And if we look at the insertions across the chromosome, we see in the parental strain pretty good coverage, as we'd expect, across the chromosome. And then in the transparent genes are those that were deleted from the AC5 strain, so they're not present in the organism. But we can see kind of as a broad overview that there are less insertions in AC5, which is exciting and what we expected. And I'll just kind of walk through a couple of examples of like unique scenarios. If we kind of just draw a bit of an arbitrary cutoff for the moment of the number of inserts that determines if a gene is essential or not at about 10 inserts per gene, making it non-essential. We can color each gene by, and sorry, this is hard to see. We can color each gene by its essentiality call, and you can see genes that now have red borders would be considered essential in AC5. They receive very few inserts, while genes that have blue borders are still non-essential. And we can see kind of as a bird's eye view that many of the genes that are on this chromosome appear to not tolerate insertions in the AC5 cell. And to just look at a couple examples really quickly, this one stands out. We have a stretch of a few non-essential genes here that receive inserts in the parental strain, but receive very few inserts in AC5. And if we kind of try and categorize something like that, we could say that this is a conversion from a previously non-essential gene to an essential gene, probably as a result of the reductions that we've made to the chromosome. And then more surprisingly is the conversion from an essential to non-essential gene. You can see very few inserts in this, the parental strain up here in black, and then many inserts in the red here. Admittedly, I'm much more skeptical of this type of conversion. We see it elsewhere in the genome occasionally, but it's possible that this is, because the chromosome is a circle, it's possible that this is the result of an aneuploidization when you get inserts in this gene. And we haven't experimentally verified things like this, but if it is true, it's very exciting to see this. And then really what we wanted out of this data, more than the conversion factors, is really what can we still delete from the chromosome? And it looks like we've got a pretty good idea now. I'm just indicating in these arrows here, the chromosomes that it looks like, the essential genes, or sorry, the genes that remain non-essential or become non-essential that we can potentially delete from the organism. So we have that chromosome, we can go in and make these deletions and test and hopefully arrive at a truly minimal version of the chromosome. And with that, I'll just say we are planning to do this same approach on 5 additional chromosomes, sort of to test the limits of this method and to kind of improve the number of genotypes that we get out. So we might be able to do genetic mapping across different variants, and then hopefully also combine these chromosomes into a single cell to work towards a minimal genome. And with that, I'll just thank my lab. Ian Ehrenreich, my PI, has been extremely supportive and awesome. It's a pleasure to work with him. And then my co-author, Kara Hull, who has been instrumental in this work. And thank you all for listening. And it's been a pleasure listening to the talks today. And I'll take any questions if there are any.
That is so cool. But one quick question that I have. So as a gene goes from essential to non-essential, have you, I mean, it makes me think of like toxin antitoxin systems that you've taken
Hmm.
out one component, now you can take out two. Um, those I would assume such things exist in yeast, but
Yeah.
when we were trying to do something similar with Chromosome marxianus, I think Ian was
Mhm,
even here then.
mhm.
We found that things became idiosyncratic. You took something out and the response made no sense sometimes. But then we
Yeah.
also lost a lot of the yeast tools when we couldn't go from yeast back into Chromosome very easily.
Yeah. Yeah, definitely. I think Ian and I have been talking a lot about this toxin antitoxin system, and we have another project in the lab that's kind of focused on looking at smaller eukaryotes and what genes are present in them, and why it might be the case that you can. So a good example, which isn't necessarily toxin antitoxin, but is genes that mediate intron splicing in yeast are essential, but that's only because introns are present in a handful of genes. But if you remove those introns, those genes are no longer essential. Um, yeah, and then to the point of the yeast machinery, yeah, that's exactly the experience that we had with BA6. AC5 is better, but yeah, BA6 we basically couldn't do any work. We couldn't do CRISPR edits in the cell or anything. It wouldn't tolerate any holding on to any plasmids either. So it's we quickly reached a point where things became very difficult.
Okay, I've got to try to keep going on, but thanks, Zach. I really like this, and congratulations. It's a beautiful piece of work.
Thank you.
And it'll be interesting seeing how you guys go along with the stuff going on in Manchester.